So to start us off, we're going to have Shaun Rawson. He's a final year PhD student from the University of Leeds, otherwise known as the place for two Titan crises up north. So Shaun, the floor is yours. Take it away.
Right, thank you very much. Thank you for the opportunity to speak here today. So I'm going to tell you about some of the work I've been doing as part of my PhD over the last three and a half years in the group of Stephen Munch, who's over there. So the object of my PhD was really to see, is EM a viable tool for inhibitor design and inhibitor discovery? So I'm sure all of you are aware that traditionally, rational drug design has been underpinned by NMR and crystallography, primarily crystallography with NMR kind of as a bit of a sideshow. So at the start of my PhD, EM for rational drug design, that was nowhere. That was a bit of a joke. So the question we had is, could EM be useful, or is it just a pipe dream? So the system that we used to look at this, primarily, was the V-ATPase. So for the purposes of my talk, all you need to know about the V-ATPase is it's big, it's a rotary ATPase, it's a motor, and it acts by pumping protons across a membrane by hydrolyzing ATP. So this section down here rotates, and that's where the protons go through the membrane. It's also an important drug target, potentially implicated in a bunch of different diseases, cancer, osteoporosis, osteopetrosis, etc. So we figured we'd have a look at this. Now, there are already a number of V-ATPase inhibitors known, including this big beast of a molecule here, bifalomycin. Now, all of these existing V-ATPase inhibitors suffer from the same problem. There's no specificity. So if you take one of these V-ATPase inhibitors, it will inhibit every V-ATPase in your body, and then you will die. So that's not ideal for an inhibitor, certainly not ideal for a drug candidate, yeah? So a couple of years ago, a group in France discovered this toxin called P-albumin 1b, extracted from peas. Now this, again, inhibited the V-ATPase and had one particularly interesting property. It only interacted with a V-ATPase from a small subset of a weevil family, of all things. So what we want to know is, how does it get this specificity? Why does it only inhibit the V-ATPases from this small subfamily? And if we can work out how it gains its specificity, can we mimic this and use it in something slightly more useful and elegant than a P-toxin? So the tool that we were going to use to look at this to begin with was negative stain. So negative stain, it's quick, it's easy, it's low resolution. So PA1B on the scale of negative stain is actually too small to see relative to the V-ATPase here. So in order to visualize it, we have to first conjugate it through biotin streptavidin to a HRP tag, just to add some extra steric bulk so you can actually see the thing in the microscope. And the advantage of this is it was very quick. All we really want to know initially is, where did this little guy bind on this full complex? So that's what we went and did. Now, at incredibly low resolution, this is the V-ATPase. It is, effectively, two blobs on a stick. So we added some PA1B with the HRP tag, and hopefully, if it binds, we will see an extra massive density somewhere on the complex, which we can then use to infer the binding location of the inhibitor. You add that in, and in your negative stain classes, you get an extra massive density at the base. Now, these are classes, so they might be a little bit harder to see. So if you look at it in a 3D model, you instead get this extra massive density at the base here in a roughly 120-degree arc around the bottom. Now that's kind of what we expected to see. So one thing I didn't mention, most V-ATPase inhibitors that are known, certainly the great majority, bind around this C-ring here. And so this wasn't overly surprising. And the action of most of these is that upon binding, they're then rotated into the subunit interface by the turnover of ATP, like this, and this is what we expect to happen with PA1B. So it's bound to the C-ring, rotates until it strikes the A subunit, and acts like a literal spanner in the works, preventing further rotation. So the obvious way to test this, you add ATP to the sample, and what you should see is a localization of the inhibitor to a single point at the CA subunit interfaces. So that's exactly what we did. And again, before addition of ATP, you have this 120-degree arc here. After addition of ATP, you get localization to a single point. That's great. That's what we wanted. However, that single point is not the CA subunit interface. The CA subunit interface would be closer to this side here. So actually, we're finding localization at a point distant to the expected mechanism. So with some collaborations with a group in Germany to do some biochemical radiolabeling, Well, actually, what we discovered is that this toxin binds to both the C subunit rings and also the E subunit. Now, this is interesting for V-ATPase because previously the location of the E subunit was unknown. Nobody had any idea where it was in the complex at all. So even with a very low resolution negative stain model, we can rapidly, so this was done in the course of a few weeks, identify the location of a totally novel subunit and at the same time show that even at low resolution, EM can be a viable tool for at least showing the binding location of a previously uncharacterized toxin or inhibitor. But that's low resolution. Nobody cares about low resolution anymore. So we thought, could we do better? The answer is, yeah, a little bit. So again, this was not taken at EBIC. This was data from the LMB. We collected a few micrographs of the V-ATPase, and we get a relatively nice reconstruction at 9.4 angstroms, so sub-nanometer. And what this allowed us to do was to accurately fit in homologs of crystal structures of each individual subunit into the complex as a whole. So this is great because it allows us to start exploring subunit interfaces which crystallography can't see. So you might get 2 or 3 subunits next to each other in crystallography and be able to probe those interfaces, but you're never going to be able to crystallize all of these next to each other and find out what's going on. So this enabled us to notice a few different things in the structure that the crystal structures of the individual subunits haven't shown. So for example, in the C subunit here, we identify this highly charged pocket at one end, which isn't visible in most of the crystal structures. So one thing we're trying at the moment is carrying out a virtual high throughput screen to identify some potential binders, and one thing that's ongoing at the moment is we're now screening them biochemically to see if any of these hits are real, but it's virtual screening, so probably not. Again, this is okay, we're at sub-nanometer resolution, it's all right. It's not really good enough to do anything close to rational inhibitor design. So the question is, why? Why are we stuck at 9? There are people with 3 angstrom structures, there are people with 6 angstrom structures, why are we stuck at 9? The answer to that is flexibility. So a few years ago, we did a study in negative stain where you show that there is a significant flexing along the long axis of the V-ATPase. And we think this might be one of the things that degrades the resolution of the complex as a whole. And indeed, if you watch the little movie of the dancing hula V-ATPase, you can see there's a significant amount of movement with the 2 domains relative to one another. So if you try and counteract this in 3D, in the cryo images, we get a significant increase in our resolution. So on the left, this is the V1 domain from the uncorrected images where we haven't done any kind of masking or anything to try and counteract this movement. And the core here is around 9 angstroms, which is okay. But after you've done this correction and you've accounted for the mobility and flexibility of the V-ATPase, we improve our resolution in this region to around 6 angstroms. So just by a few simple tricks and steps, you can get a 3 angstrom gain very easily. But even at 6 angstroms, any crystallographer will tell you that's nowhere near good enough to do rational drug design. So we thought, yeah, other people can do high resolution structures. Can we? So last year, the first few structures coming out showing inhibitors bound in EM maps directly, such as the beta-gal structure, TRPV, and the proteasome structures. So we thought, could we do this? And if we can, we're probably going to need a different specimen, because the V-ATPase clearly is not ideal for this. So for this, we move on to a new test system called IGPD. Again, I'm not going to tell you anything about this protein. So it's a histidine, it's involved in histidine biosynthesis in plants. It's a potential herbicide target, which is why a few industry groups are interested in it. But the reason we chose it is it's really easy to get hold of, it's abundant, it's very highly symmetrical with octahedral symmetry, and it's an absolute brick. So if we were going to get to high resolution with something, it would be this. So because we don't currently have access to a high-end microscope, I flew to China and collected a little bit of data for it. And to cut a long story short, we got a pretty nice structure at the end of it from a relatively small number of micrographs. So you can see you get well-defined classes with clear structural information and secondary structure in the slice-throughs. Local resolution suggests that the map extends down to around 2.4 angstroms in the core, and you have relatively nice side chain density in some areas just by eye. This enables us to do a de novo build for the entire protein backbone into the structure based on nothing apart from the sequence and the secondary structure prediction. That's 2 weeks of my life I'm never getting back. This then enabled us to perform some this allowed us to then do refinement in RefMac to produce the full structure. And again, if you look at the side chain densities after this refinement in RefMac, you can see that actually it's really rather nice, like you'd expect from a 3.1 angstrom map. So most of our side chains are well resolved, everything looks pretty nice. But what about the inhibitor? Now, if a crystallographer wants to find an inhibitor binding site, the first thing they do is do a difference map. And if it's good enough for a crystallographer, it's good enough for us. So this density here, shown in green, is the density you get when you subtract the model that I built from the experimental density. And you can see there's a clear green lump of density, effectively, which corresponds well in shape and size with the inhibitor we know is added to the sample. So that's quite a nice start. Then if you redo the refinement in RefMac with the inhibitor added, and also these manganese ions that we know are important for the activity, and we know are present in our sample, you can see that actually you get a very nice binding site where everything fits in pretty well, everything is in the right place, and all the bond distances are approximately correct. So this is pretty good validation that actually EM can directly visualize an a bound inhibitor in a protein. So maybe there's hope for us yet. And just to compare, so one thing you never show over VM is your map real? Do you have any confidence that what you've got is actually there, or is it an artifact? So in order to validate that, we compared it to a 1.1 angstrom crystal structure solved by our collaborators in Sheffield. So overall on the protein backbone, we have a very good agreement with a decent RMSD between the two structures. And there are some minor differences in loop regions and the terminal regions, which could be due to differences between crystal packing artifacts, the different environments that the two samples were when they were imaged. If we zoom in to the inhibitor binding site specifically, you can see, again, very good agreement, particularly in the side chains flanking the inhibitor itself. There is a subtle difference with the inhibitor bindings with the X-ray structure shown in gold. If you notice, the X-ray structure doesn't actually seem to fit that well, the inhibitor there. We don't know why this is. I mean, the most likely reason is simply a resolution. We're at 3 angstroms, the crystallographers are at 1 angstrom. Chances are we just can't resolve the positions as accurately as they can, and that would explain these slight differences around here. But there's also a bunch of other reasons it could be. So, for example, there's a number of negatively charged side chains just shown at the back of the image. And in the EM density, these are shown to be preferentially damaged. So actually, the difference in radiation damage between the crystal structure and the EM structure could be changing the location of the inhibitor. So even though we can see it, we don't know for certain if that's where it actually is before irradiation. So it's something to be aware of. We don't know if this is a potential benefit of EM because we get our structure in a more native conformation without crystal artifacts, but maybe it suffers from radiation damage more strongly than the corresponding crystal structure. So to conclude this little part of the story, IGPD has been useful as a test specimen to show that we can use EM to directly visualize inhibitor binding in a complex, and we can then use this to inform the design of better, more potent inhibitors based on this information. This system was ideal because it allowed us to also validate our information against a well-known, well-studied complex with a high-resolution crystal structure. And I hope I've shown you at least that EM can be useful for this. There is possibly some role in using EM to directly show inhibitor binding. And indeed, not just at high resolutions. So even at low resolution, EM can show you very quickly the location of an bound inhibitor. At moderate resolutions, you can use existing crystal structures and homology models to explore new interfaces and look at different ways of inhibiting those or the formation of the complex. And then at high resolutions, we can now directly visualize inhibitors in exactly the same way a crystallographer can. And if crystallography is underpinned rational drug design, maybe we're next. So I'd just like to thank all my supervisors at Leeds, Steve, Robin, and John, Mike Harrison and Claire for help with the V-ATPase work, Dan Hurdis, who will be somewhere in the back there, who made most of the figures for this talk, so thank you for Photoshop, and the University of Osnabrück for providing the V-ATPase samples, and the University of Sheffield for the IGPD samples and their help with the crystallography. And you for your attention. Thank you.
Thank you, Shaun, for a lovely talk. Do you have any questions?
So I'm a crystallographer from industry, so I've got a real keen interest in how EM develops over the next few years and its potential scope for pharmaceutical development. You've obviously demonstrated one example here. So firstly, how potent was that phosphonate ligand? And secondly, how many other ligands have you tried?
So the ligand we chose was, again, one of the reasons we chose this system. It was a nanomolar affinity ligand, so chances are we'd have high occupancy and it would make our life easier. If you're going to pick a test system, you pick the easiest one you can. So we haven't tried any other ligands yet, so other groups have found some issues where the occupancy isn't full, and that has issues with the averaging, so you don't get complete density of the inhibitor.
And then perhaps a follow-on question, how long did it take you to achieve that structure?
This structure, so the data was collected in less than 1 night's data collection, and the processing for the initial map took around a week, week and a half, and then to get to the final end map, a long time, several months, but that's because we were learning along the way effectively. I'd never built a structure before by hand, so the process of building it in Qt was painful. I imagine it'd be a lot quicker for a crystallographer.
Don't count on it.
Ed?
Hi, Shaun. Very nice density for the ligand and for your metals, but it can be quite tricky to identify what metals you've got just from density. That's one of the advantages of crystallography, I guess. Can you comment on how you might identify metals if you really didn't know what they were?
So for this sample, we have the advantage that there's quite a lot of biochemical evidence already on this sample. So we know that manganese is involved in that pocket. We know that manganese is crucial for the assembly of this complex from the monomer. And, well, we know there's a hell of a lot of manganese in our buffer. So for this, manganese was a pretty good guess for what it would be. And actually, if you contour the map right down, the 2 strongest peaks remaining in the density correspond with the location of those metals. But you're right, the actual identity of the metals is not something we could unambiguously assign without other biochemical information.
Okay, in the